Welcome to the Ashby Village Science and Ideas Group. I'm Joe Evinger, and I coordinate this group's events with the help of our co-host and tech expert, Hillary Naylor. All of our members here have very educational and work experience backgrounds, but we all share a common interest in science and new ideas. We currently have over 70 members in our group, and another 100 or so on our guest list who occasionally join us. We meet the second Thursday of every month at 3 o'clock until about 4:30. We have a different guest speaker each month who speaks on a topic related to science and ideas. We rely on our group members, all Ashby Village group members, our invitees, presenters, and others for ideas about new speakers and ways to connect with them. I want to take a minute to go over some future speakers between now and the end of the year. On July 11th, we're going to take a different direction. Mary Mackey, who is a PhD in comparative literature, will talk to us about creativity and thinking outside the box. In August, Mary Kawar, I think she's joining us today, an occupational therapist, is going to talk to us about neuropsychology of the vestibular system. And I admit I immediately looked that up in Google to find out what that meant. To my knowledge, it's about the inner ear and keeping your balance and having a sense of where you are spatially. This, of course, if she has some practical exercises, perhaps she will discuss with us to enhance our own vestibular processing so as to help mitigate the physical, mental, and emotional decline that often accompanies people in the aging process. In September, Psychology will join us, and their mission is to protect threatened island ecosystems all over the world. In September, a new person I just signed up, Smithsonian curator Matt Schindel, has released a book, For the Love of Mars, A Human History of the Red Planet. Recently, I've sent an invitation to someone who's just written a book about poisons, good and bad, for all you Agatha Christie fans. We hope to get him to speak to us next year. We're always looking for new speakers. As I said, we're completely booked for the rest of this year, but open for next year. If you know someone, it helps to drop a name, as I've done today with Dr. Flint. Please get in touch with me about that. We invite everybody to join, Ashby Village members or not. I do have to note that 501 is Ashby Village is a 501c3 tax-exempt profit organization. I asked Hillary to place a link to the website in the chat. We rely on partially on donations to continue groups like this. Today, Dr. Jonathan Flint will present a talk titled The Genetics of Behavior. Sarah Evinger will now introduce Dr. Flint. Sarah?
There we go. Dr. Jonathan Flint is a psychiatrist and geneticist who, until 2016, when he moved to UCLA, was a professor at the University of Oxford in the UK. He's now one of the leaders of UCLA's Depression Grand Challenge, which aims to identify the causes of depression and use that information to design new treatments. His research is focused on understanding the genetic determinants of behavior using both animal models and human studies. Most recently, he has worked with colleagues in China and Korea to recruit participants to studies that aim to identify the causes of depression using genetic strategies. Dr. Flint is also involved in work that seeks to use voice recordings and what are called digital phenotypes, which is the data collected from cell phones and wearable devices, to identify changes in mood. So we'll be sure to ask you about mood rings, Dr. Flint.
I get off of this. Am I off?
Hillary, I think if you need to silence someone here.
So ready to start?
Yes, we're ready to start, Dr. Flint.
Okay, let me just bring up the PowerPoint. Okay, thank you very much for having me. It's a pleasure to talk to you. I'm going to be talking to you for about an hour on the genetics of behavior, and specifically around the work we've done on the genetics of depression. And as you see by the subtitle I put in here, there's an issue around how we actually establish these findings. And I've divided this talk into two parts. In the first part, I'll be describing some background and the difficulties that we in this field have faced. And in the second part, I'll be talking you through a study that I've done, trying to give you a feel of what it's like, as it were, in the field to carry out this kind of work. So I'm going to start in 1996, and here's a paper with I assume to most of you, a fairly incomprehensible title. And I'm going to talk you through what these components mean and why it was important to us. And essentially, there are two things to notice here. There's something called anxiety-related traits. And then there's this longer term, the serotonin transporter gene regulatory region. And the last thing is this thing at the very beginning, the association. So let's start with the anxiety-related traits. What does that mean? Well, in this context, it really relates to a personality trait called neuroticism. And you can think of that as being like an exaggerated version of Woody Allen, someone concerned about small things, like whether they have cancer because they see a black mark on the back of their hand, and constantly worrying about future problems that may occur to them, but seem to the rest of us to be not particularly important. And personality traits are typically measured by something like this. Self-scoring personality tests. I bought this in a train station, I think, in Washington, D.C. So to give you some indication that this is of general interest. Most of us like to know something about our personality. And the person who wrote this, Viktor Serebriakov, makes sure that we realize he's very important by saying that he's the honorary international president of Mensa, that society for people with extremely high IQs, to assure you that you're buying something here which is valuable. So let's open it up and see what the questions are. So some of these questions you can imagine are telling you something useful. Would you like to be a lion hunter? I'm not quite sure about that. Do you like to pick up furry animals? What on earth is that one about? And if you notice this one here, number 114, do you disregard other people's feelings? And I'm sure most of us are going to have difficulty with that one because it rather depends, doesn't it? I mean, you know, like if I'm really trying to impress you, take you out for a meal on something, I won't disregard your feelings. But if you're standing in the way of me and my drink of Chardonnay at the end of the day, or a glass of beer, yeah, I probably will disregard your feelings. I sort of prioritize sometimes. So it's a little odd, I think, for us to realize that's not quite what the psychologists want us to think when they give us these questionnaires. They're really asking us to just give a yes or no brief answer, and they're not too concerned about whether we think that the answer is true or not. And to give you a specific example of this, let me show you a questionnaire that we used. This is not Serebriakov's. This is something called the ISENC personality questionnaire that we've given out to tens of thousands of people to assay these anxiety-related traits. And you can see we're again asking questions, some of which you may recognize as being related to your mood. Number 41 here. Would you call yourself tense or highly strung? Yeah, you can see how that might be related to people's mood. But there are other questions here, like this one here. Is or was your mother a good woman? And we're asking you to say yes or no. And the people involved in our studies sometimes have some difficulty with this. So question 61, I found this very hard to answer. My mother was good in that she provided us with clean clothes, food, and tidy and clean house, but she was and is emotionally cold and lacks empathy. I can understand, but since warmth and empathy is so essential to the well-being of children, I found it hard to deal with her now, as over time her cruelty seems to have increased. She had a hard childhood and years of abuse from my father. It's not surprising, but hard to say whether she is good or not. Now, that is not a yes or no answer, and that's not really what we need. We're not about these yes or no answers. We just want you to say, just simply tick the box. So why would we take this very simplistic view to what are these rather complicated questions? And the answer is that if we do force you, as it were, just to say yes or no, we find that there are patterns in the data. So here are two questions. Are your feelings hurt? And do ideas run through your head so you cannot sleep? Now, there's no logical reason why, if you answer yes to one, you should answer yes to another. But I promise you, if I ask those two questions of this group, on average, people who say yes to one will say yes to the other. In other words, there's a correlation between these two, regardless of whether you're telling the truth or not. We find these patterns. And if we look through all of the questions in those questionnaires that I've shown you, these patterns emerge. And we can use a statistical analysis which will extract what are called factors. And this, in essence, is what we mean when we talk about neuroticism. It's a correlation between the questions that we give you in that questionnaire, nothing more than that. Now, you might think that might not tell us very much, but there's been a lot of work looking at those factors. And one of the things that they predict is, for example, whether you're likely to suffer from an anxiety disorder or whether you're likely to suffer from depression. So now I've told you a little bit about the first item in that paper, the anxiety-related traits. You know it's about a personality trait, and you've got some feeling about what it means. Let's move on to the second item. So this is a bit more complicated, the serotonin transporter. Sorry, I'll come to that one in a second. Good, I have my slide advancement here. So the second thing I'm going to just want to deal with is this business of association. So this is a genetic term. So what does this mean? And I can simplify it in the following way. I hope you will know you have one copy of DNA from your mother and one from your father. And I've made a very simple DNA schema here. Obviously, in practice, you have many more bits of DNA than this. But occasionally, the two copies differ. So if you can see, I've highlighted in red here that there's a G, say it's from your mother, and a C, and that's from your father. And because we have a G and a C, and we can randomly pick any combination of these, the consequences are that there are three possibilities. You might have two Gs, you might have two Cs, or you might have, as shown here, a G and a C. So make it nice and simple like that. And in an association, a genetic association, we want to know whether the people who have the GG, GC, and CC might differ in some trait. For example, they might be severely affected if they have the GG with anxiety, and might be moderately if they have the GC, and unaffected if they have the CC. And we can change that from an affection status to a quantitative measure like height. So the people with GG might be the tallest and the people with the CC the lowest, and so on. So that is the essence of genetic association. It's the second component. And the last component, the more technical here, is this thing called the serotonin transporter. And that's a gene, and that's indicated by this little figure here, whose complexity you don't have to worry about, except to realize that this part of the gene, is the front of the gene, is a regulatory region. That's to say it controls the amount of product of the gene. And there happens to be a small change in the DNA, like the one I showed you before, a G or a C. The only difference is that this is a little bit missing or a little bit inserted, depending on your point of view. And that means that you can produce, as we've had before, these differences. So we have some people might have a short with S, and some people might have a long version with L. And just as I created three genetic types with the G and the C, I can produce the same with the S and the L. So people can be categorized as whether they're two Ls, two S's, an L and an S, and I can then carry out the genetic association. And in this case, I'm going to use my measure of neuroticism, my anxiety-related trait. And I'm going to give a lot of people this questionnaire and see what they score. So here are the results. This is from the paper I showed you at the beginning. This is the number of people, and this is their neuroticism score. And you can see that there's a difference. The people with the LL have a lower score than the LS and the SS. Now, the people who wrote this paper decided, because these two numbers are pretty much the same, that they could just combine them. So they end up with a picture like that. And then they run a statistical test to see whether there's a difference between those two scores. And the answer was, yes, there is a statistical difference. Now, this paper was published in 1996, and now in 2024, it has more than 6,500 citations. That's to say, other people have been quoting this paper many times. And to give you some feel as to what that number is, I think that the median citations for a publication are somewhere around like 1 or 2. Most papers that get published don't really get read very much. So I'm happy if my PhD student publishes a paper which gets 10 or 20 citations, very happy if they get 100, and it's extremely unlikely that I'll get anything like 1,000. So this, on the face of it, looks like an enormous success, hugely cited paper indicating a very important finding. Unfortunately, that turns out not to be quite true. So in the few years that followed the publication of this paper, a number of people tried to do the same experiment to replicate the findings. It's relatively straightforward. I'm going to need maybe 100 people. I'm going to give them this questionnaire, and then I'm going to test for this genetic variant. And here's a little summary. I've just took at random 4 papers, and this is the number of people, and this P is the p-value, and if it's less than 0.05, then it's a significant result. And you can see that this person published a paper which was significant, and then there are 3 which are not. So this was the situation up to about, let's say, 2002, 2003, when I met Marcus Manuffa, who came to be a close friend of mine. So Marcus is a psychologist, and he's Sicilian by extraction and has a slightly pugnacious manner. He works for the territorial army in the UK. You can go off and fight, basically, on the weekends for the army. This is the sort of thing that Marcus would do. So he was slightly more aggressive in some of his approaches than I was. And I was sort of discussing this with Marcus, and he said, well, you know, so there are a lot of different results here. Why don't we apply a technique to see who's right and who's not right? And I said, well, how can we do that? And he said, well, we can combine all these studies, all the bits of information out there, and then come to a conclusion. And he introduced me to something called meta-analysis. And I'll briefly show you how this was put to me by Marcus. Marcus said, go out and collect all of the papers. So this is a list of the papers that were published, and get the data from those papers and put them in a format that looks like this. So there's a vertical line here, and think of that as being 0, means nothing. And each study is now represented by a horizontal line with a little tick in the middle. And the little tick is the estimate that they have as to whether this genetic effect increases your neuroticism score or decreases it. So if you're to the right of the line, you'd be increasing it. And if you're to the left of the line, you'd be decreasing it. And then the horizontal line here represents the likelihood that it's significantly different. So if the horizontal line doesn't overlap the 0, that turns out to be a significant result. And by putting everything into this format, what it means is you can add everything up, and then you'll get a little line down here and see whether the sum of all of these results is significant. So we did that. So we summed up all the evidence of the association between neuroticism and the serotonin transporter, and the result was the following. We found nothing. No evidence of association. So that was a little surprising. But we went a little further than that. We decided, since I was involved in another study also looking at neuroticism, to specifically test it in our own sample. So let me just briefly show you how we did this. I, at the time, was living in Oxford, which on this map is to the right, just off the map here. And what we were doing was we sent out questionnaires. This is a self-assessment, so we could send out questionnaires to all of the people in the west of England. We got their names and addresses from local doctors. And we wrote to them and we said, we're interested in studying the relationship between neuroticism, anxiety, and depression. Please fill in this questionnaire. And we got back a very large number that looked like this. which is a large number of results, 88,000. And you can see there's a distribution. So this group here will be the very highly neurotic, and this group here would be the not neurotic. And our plan was then to write to these two groups and ask them to give us a cheek swab so we could extract the DNA and run our genetic test. And mathematically, we worked out that if we just sampled from those two extremes, we were getting almost all of the genetic information that this enormous number contained. So we wouldn't have to look at all 88,000, we could just look at a few thousand. So that's stating it simply, as I would as a scientist. But there was another way of thinking about this, which was I hadn't really thought about, which is we're just about to write a letter to the most neurotic people in the west of England. Like they're on the, 3 standard deviations. They're like really, really neurotic. So what are they going to say when they get a request from Dr. Flint for a cheek swab? I hadn't really thought this through. So I was a little surprised when some of these people actually bothered to write back to me. So this woman wrote back and said, I've just received your request for a mouth swab. Beg your pardon. This is the last straw. My holiday has been cancelled. My son has influenza. My husband has left me. Now this from you. It is the end. So that reassured us that we were indeed identifying highly neurotic people, but it did mean that I wasn't going to necessarily get all the sample giving us what we want. And if you look at what we did receive, here's the distribution. This on the right-hand side is the neurotic group, and you can see less of them responded. And in a sense, that's what we'd expect. So we'd collected the sample, and then we carried out our analysis. What did we find? So this is the results, and I've divided them up by male and female. The score on the vertical axis is their neuroticism score. The first thing you notice is that women are more neurotic than men. That's what the scale tells us. The second thing you notice is I've divided them up by their genetic markers. So SS, the short, LL, the long, SL, the short, long, the mixture of the two. And I'm looking for differences in these bars. There aren't any. You don't need a statistical test to see that. We saw nothing. So that really confirmed the results we'd seen from our meta-analysis. But, we're scientists, we're deeply neurotic and obsessional, and we really want to get things right. And I had a colleague in the east of England who'd done the same study, but this time from 30,000 men and women. And we asked them if they would do the same genetic analysis. They had 20,000 men and women. We selected again from the extremes for genotyping, and we got exactly the same result. There was no effect. So I thought that was science in action. Someone had published a paper that we worked carefully through it. We were pretty certain that there was no effect. Now, while we were doing that, other groups, of course, were doing research. And within a few years, this paper had appeared. It is now at 2003. So we've now moved 7 years on from that first paper. So you'll recognize maybe some of these terms. So this 5-HTT, that's the serotonin transporter gene, the same thing I was talking to you about before. So exactly the same genetic thing. And we've got depression. It's, as you'll see, really the neuroticism score again. It's susceptibility to depression. But the difference here is now this term life stress and moderation turns up. So there's something different about this. So what was this paper all about? Here is the key result they published, which got a huge amount of interest. So now this is the vertical axis, and think of this as my neuroticism score, my likelihood that I will get anxious or depressed. And on the horizontal score, we've now got a measure of whether you've had a bad thing happen to you. This is particularly about childhood. So no maltreatment, parents looked after you well. Probable maltreatment, didn't let you watch TV when you wanted to, didn't let you go out at night to parties, and this is very bad things happening to you. And here are the 3 genetic markers I'd mentioned to you before, the long, the short, and the short. And the distance between them indicates whether there is a genetic effect on the probability of depression, increase in neuroticism. So here, when there's severe maltreatment, there's a big difference, very marked. But when there's no maltreatment, these three groups coalesce. There's no genetic effect. So the implication is you only see the effect of this when there has been some form of maltreatment. And this was called a gene by environment interaction, or that your genetic effect depends upon the environment. The susceptibility depends upon the environment. The psychologists loved this finding. It seemed very natural to them that this should be the case, and he would explain why the earlier study I showed you hadn't produced any results, why there was discrepancies between findings, because people hadn't measured the environmental impact. So we sort of heaved a sigh at the appearance of this paper, and I went back to my friends in the east of England. Remember, they'd collected the neuroticism questionnaire, this susceptibility to depression, and it turned out they had also collected for their 20,000 people a life events questionnaire. So we could try and replicate the study that had been published in 2003. And we attempted to do that. And our conclusion, as written here, is that we couldn't replicate this finding. And of course, we were not the only people trying to replicate. Many other people did this. And by 2008, there were 33 of these studies, some finding one thing, some finding another. So I can guess that you know what I'm going to tell you next. We carried out, with my friend Marcus, a meta-analysis. We put all of the results together, and you may not be surprised at what's coming next. We didn't find anything. There was no evidence of a significant interaction. So all this is taking time. It's now 2009, and I got an invitation to go to a conference, I think is in Arizona, and met this person here. This is Kathy Mary Kangas, who works at the National Institutes of Mental Health. So she's a psychologist and a geneticist. I know her work, not met her before. She sees my name badge and she sees the name badge of Marcus, and she comes up to us both, say, "Hey, you guys, I really want to talk to you." And she puts her arm around us, which of course, as English people, we do not like. Please just be polite and introduce yourself. And the reason she was so excited and wanted to see us, because she'd been working with her colleague, Neil Risch, and they'd done exactly the same as we'd done, and they'd run a meta-analysis, and they also had found nothing at all. And they'd published this, and this generated quite a lot of interest in the scientific media. And Constance Holden, who was then a journalist at Science, wrote this piece entitled "Back to the Drawing Board for Psychiatric Genetics." I got hate mail because of this. How could you not find this thing, Dr. Flint? You must be wrong. The whole thing had become really quite polarized. And the paper that we wrote about, the one that neither of us found an association, a replication for, it's continued to be cited. And I looked up recently, this is 11,000. This is twice as many citations as the other paper. Oh, okay. So giving this really as a sort of introduction to the study of behavior by geneticists, and to give you a feel that the field has been a little contentious, and we haven't really known how to solve this problem. And I'm now going to turn to look at what we think the answer is and how to carry out those sorts of studies. And there's nothing, I think, really fundamentally different from what you've been told up to now, apart from 2 points. So I'm going to introduce my next colleague. This is Peter Donnelly. He is a statistician. He is not a biologist. And when he came into this field, he knew nothing about genetics. He'd made his name working on probability calculations. And the reason that's a good thing is because he didn't have an axe to grind. Peter came at this without any presuppositions as to what was right or wrong. And he was not going to write me hate mail because of the statistical analysis we'd run in a meta-analysis. No, he was just going to approach it as a statistical problem. He doesn't really care whether the phenotype is depression or neuroticism. He just wants to know whether we're going to get the answer right. So handing over the analysis to a different group was a good idea. And the second thing is that the analyses that he was running are what are called genome-wide. That is to say, rather than looking at just one gene, the serotonin transporter gene, he was running a technology that could look across the entire genome, every single gene, every bit of your genome, all at one go. And the reason this worked was for the very simple reason that he had access to a very large sample. Just as I was talking about the 88,000 when we extract information, that's the same approach. And Peter, being a statistician, was able to work out the sample size we might need, and it turned out to be much larger than most people had been considering in the thousands. Let me just show you exactly how this works. So this is a result from the key paper. This is now 2007 on Crohn's disease. And what you're seeing here on the horizontal axis, these numbers are the numbers for the human chromosome. So we have 22 what are called autosomal that we all have, and an X and a Y, the two sex chromosomes. And the vertical axis is the likelihood that the association is true or not, whether it's correct. And these little dots here represent the result from, for simplicity's sake, just call it like a gene from a single gene. It's not quite like that, but we'll come back to that in a moment. But it's the result from a single gene, and you have a lot of them, about 20,000. In fact, this is summarizing result from about 500,000 tests. And the reason we're getting these blocks of blue is simply because there are now so many little dots that all the colors coalesce. And there are regions in the genome where the little bars protrude. And they're marked in green here because Peter thinks that these are significant and we should take them seriously. And because there are a number of these sticking up, he called this a Manhattan plot on the assumption that it looks something like the Manhattan a skyscraper view of New York. And these are highly significant. So this is 10 to the -5, 10 to the -10, 10 to the -15. And for technical reasons, we think that anything over about 10 to the -7 is likely to be true. So that was for Crohn's disease. I'm going to show you next the full results from this paper. So notice it's 7 common diseases. I'm just showing you one. So this is the result from all 7 diseases, and you can see that we're getting these nice tall green peaks for type 2 diabetes, rheumatoid arthritis, coronary artery disease. And notice as your eye runs over this figure that there's one at the top here, which is a psychiatric condition, bipolar disorder. And that's not looking so, green. It's not looking so great here, So the genetics of hypertension, Crohn's disease, well, hypertension is not great. There's something here, but certainly rheumatoid and type 1, but bipolar is not as good. I think you'd agree. And just to make that point even more clearly, here's another study from a couple of years later, another 3 Manhattan plots. You can see these very clear skyscrapers indicating positions on the genome, which increase the risk of Alzheimer's disease, coronary artery disease, and type 2 diabetes. And on this graph, we have a little dotted line here indicating the level beyond which we think something is significant, just to make life easy. And I put in the middle of this figure, major depressive disorder. Now, I've educated you all to interpret these graphs. You know that anything that sticks above this dotted line is significant, and you know that these numbers represent chromosomes. So now I'm going to show you the result for major depressive disorder as it stood in about 2010. Notice anything different? Would you say we'd found anything there? Anything sticking above the little dotted line? Doesn't look like it, does it? So this sort of raised a lot of conversations, and this is my close friend Ken Kendler, and we'd go for walks. He lives in Virginia, so we'd go out into the mountains and go hiking, and discuss problems like this. And I'd say to Ken, why isn't this genetic stuff working for depression and schizophrenia and bipolar? Is there something different about the genetics of behavior? Why would they find things for Crohn's disease and not for depression? So we talked about this and decided we should try and do a study ourselves. And we worked out that one of the things might simply be no one had got enough patients. Like maybe 3,000 wasn't enough. Maybe we needed 10,000. Maybe we needed 20,000. No one really knew. But maybe it was just that. Maybe it was just simply that. And maybe also it was because certainly for depression, we needed to be pretty careful about the diagnosis, because you can be depressed because, your television show was cancelled that you wanted to see this evening and you're feeling a bit miserable, or you can be really depressed because your partner died. Now, I'm going to treat one, but I'm not going to treat the other. But if I come and ask you how you're feeling, both of the people are going to say, I'm feeling depressed. Those are not necessarily the same conditions, and maybe we need to be a bit more rigorous how we recruit our cases. So we decided, this took a bit of time, of course, to go and work in this country. This is Shanghai in China, because it has a very large population. Shanghai itself has 20 million people, so we were pretty certain we would get the number of cases that we need. It shouldn't be hard in a country of this magnitude to identify sufficient cases. And the other great thing about working in China, it has a very good health system. There are lots of well-trained doctors, and we could work with them to identify the cases that we would need for our study. So we set up what we call the Converge study, and sitting down, Ken and I had come up with this acronym to summarize the relationship between, as it says here, China, Oxford, and VCU, Virginia Commonwealth University. We set up the Converge study, and we decided to study women only because women get depression more frequently than men. Those we wanted to have recurrent depression because we felt that was a more severe form of depression, and only collect cases who are in hospital, again, to try and identify very severe cases. And we started this in 2008. We got a group of doctors in hospitals across China, and Ken and I would go for a few days. Ken would give some didactic lectures on how to make the diagnosis and the aims of our study. And then we work with the doctors and we train them how to interview. So you can see here, each doctor has a laptop that was purchased for the study, and they're reading from our list of questions. We want to make sure we're asking exactly the same questions of all of our patients, and we record all the interviews. We check how people were going. We call them back in for a second training. Spend a lot of time being very careful about how we made the diagnosis. And we recruited something like 60 hospitals from the far north in Harbin right down to the south of China. And I'm just going to walk you through just a little bit now what it's like, because I think I've probably been to more psychiatric hospitals in China than any person on the planet. And what we would do is recruit the hospital, train the doctors. That would take a visit from me and from Ken, and then we'd follow up. So I'd go and meet the team, the doctors in each hospital that was involved, discuss with them how the project was going, see if they had any problems, if anything had emerged that might be holding things back, perhaps troubleshoot around some of these issues and potentially come up with some solutions. And as you can see, this was a little hard work, I was finding this pretty wearing. It took about 4 years to run all of this, but we did eventually collect the sample that we needed. So we were up to, in the end, 6,000 cases and 6,000 controls. And this is a graph to show you the progress that we made. This is a graph very dear to my heart, because each week we check and my heart would sort of be running a bit fast, depending on what the results would be. And you can see some slightly interesting things here. So you notice that there is these horizontal lines occurring fairly regularly across this graph. So these represent national holidays in China. So each new year, they'd all go on holiday. Nothing would happen. Times of great stress for me. But we then pick up occasionally, and you can see that the graph sort of comes a little bit more vertical at each stage. You can see at the beginning of the study, we were not doing very well at all. And if we carried on at that rate, we would have taken about 15 years to finish it, which is why, of course, I had to go over and help them and encourage them and get things done. But, here we were. We managed to collect our sample. So I've run through 4 years of my life. And I'm now going to show you the results. And I'm hoping that with the education I've given you in interpreting the Manhattan plots, you'll be able to tell me whether I was successful or not. So chromosomes on the horizontal scale, likelihood on the vertical scale, and I put a little red line here to indicate whether we've got anything that's significant. And there are just 2 places. Can you see? Yeah, just did it. Just by our fingernails, managed to get over the hurdle to be significant. So that took a while. And of course, we're not the only research group doing this. And things have changed since then. And I want now just briefly to show you the types of results that have been produced by other groups around this and other condition. And now I think these graphs should be pretty easy for you to interpret, and you'll get some feel as to what the state of the field looks like. So this is one for bipolar disorder. Look, there are many more peaks here. This is the red line for the significance threshold. The people who wrote this paper decided to write these little incomprehensible symbols up here, and letters refer to genes, positions on the genome. Don't have to worry about that. The reason I'm showing you is to say, look, they find things above this red line. Here's one for schizophrenia, highly significant. This was a very great success. There's something on chromosome 6 here, which is highly significant. Lots of people have found that. This is one, of course, for neuroticism. Turns out that there is nothing around the serotonin transporter gene, as our earlier findings suggested, but many things elsewhere. There's lots of other findings right the way across the genome for neuroticism. This one is for cannabis use with a single effect here on chromosome 3. Now, I've shown you so far sort of drugs, a personality trait, and you might begin to wonder, well, what about other sorts of behavior? And of course, many people had the same thought and were collecting, for example, here's years of education, very simple measure. How many years of schooling did you have? Did you leave school at 16, at 18, 20? Did you carry on to do a PhD? And this, like you can see here now, there's like almost everywhere in the genome is above this little line. And that is because if you read down here what they've done, 3 million individuals. And you might wonder how they could possibly collect that much data. Well, the answer is that this information you can now obtain from biobanks. And it's quite easy. Anyone with a laptop now can log on and get this information. Something called the United Kingdom Biobank will give you 500,000 data points. The US is something called the All of Us study. That's another 500,000. And other countries have been doing this for a while. So these data are now become available, and the genetic information with them. And therefore, sample sizes of this magnitude are relatively straightforward to collect. And now you can map these things like years of education. This is a measure of risk. Like when you drive your car, are you taking risks when you overtake, or are you a careful driver? Do you like betting? Things like that. A measure of intelligence. An IQ test has been mapped. Sleep, are you a morning person? Do you like having a nap? Here, look, there's a genetic effect for napping here, of all things. All sorts of stuff, all of behavior really has now been analyzed in this way, and large amounts of data have been collected for the genetics of behavior.
This is where I could use the hearing aids, make it louder.
So just to summarize and give you some feel as to what we know now, I'm going to call it what I call 4 observations. So I've used this acronym, Genome-Wide Association Study. That's just describing the results from the Manhattan plots that I've shown you, because we're screening the whole genome, and it's an association study. So what are the important findings? And the first one, simply put, and you probably guessed this, any behavior can be mapped if you have a large enough sample. And we do have large enough samples. We have sample sizes in the millions, and it won't be long before we have sample size in the tens of millions. Almost all metropolitan hospitals are now introducing biobanks. They're offering genetics analysis. In UCLA here, we now have 140,000 people who've had their entire genome sequenced. This is being rolled out as a tool for what's called precision health, that we can target treatment to the groups who need it and so on. So genetic information on an enormous scale is being collected throughout the world. And as a consequence of this, we can take that information and we can map all the things I've shown you and more besides. So it's really transformed what scientists can do. What it means, however, is something that we can argue about. So the first question is, first point is that really we can now map anything. The second point is a little bit more difficult, and I've sort of obscured this for you, really. I've written it here. These association studies do not find genes. GWAS do not find genes. What do I mean by that? I talked about genes in the first introduction to this approach. So here I'm showing you a sort of zoomed in version. And let's start at the bottom here. So these are genes, the names of genes. LHPP, FAM53, this is the name of a gene. And they're shown in this little stick cartoon as having these sort of horizontal lines with little vertical blocks. And this represents the difference between the part of the genome of the gene that's coding, as to say, the bit that's turned into a protein, the bit that does work, and the bit that doesn't, as far as we know, do anything at all. And on the top of this figure, I've got this plot. These are the results of the association analysis. So each is a little genetic variant with the likelihood of the association. You can see there's a peak here. And if you run your eye down, this peak lies in the middle of this gene, but not within what I called the coding region, not within a region of the gene that really does anything. So it could be something to do with this gene, but it might not be. And this has been a problem with these studies, that they say it could be this gene, but it isn't this gene definitely. We could say it was definitely this gene if the peak coincided with one of these little vertical ticks here, the coding regions, but that almost always is not the case. So this point gets a little, we tend to ignore it, but it unfortunately is a bit of a fly in the ointment for people who want to use results like this to try and get at the biology. Because if I wanted to understand what causes depression, I really want to know which genes are involved. So the technology for mapping doesn't give me the genes. It gives me these little bits of the genome which do something, but I don't know what they do. They might be part of this gene. They might not be. We have a lot more experiments to do. So the second point about these studies is that they do not find genes. They find bits of the genome that are related to the condition, but we don't really know what that relationship is, at least not yet. The third thing is that the individual effects that we're seeing are small. That is to say, if I go back to this slide here, we're saying there's a genetic effect here. It's let's say increasing the risk of schizophrenia. There's something here. If you have a genetic variant here, the risk of schizophrenia will go up. But by how much? If you have this, will it double your risk? Will it make it 4 times? Well, the answer to that question is unfortunately only a very tiny amount. So there is one place in the genome for schizophrenia would increase it by 1.26-fold or something like this. But almost all the other ones, all the ones down here, are a very small increment. So most of them increase it just a little bit above 1.07, 1.06, something like that. They really don't increase the risk very much. So they're useful in aggregate, but again, trying to understand what an individual locus does, what an individual effect does, is proving rather hard for us to understand. And then the next thing is that when we study these conditions in different populations, we often find very different things. So this is a result for schizophrenia. I showed you this figure before, and I pointed out to you this very big effect here, if you remember. This is on chromosome 6, on a study carried out in European population. And then a group in China did the same thing, but in a Chinese population. And if you look here, chromosome 6, 1, 2, 3, 4, 5, 6, nothing there. So here's 6 in Europe, here's 6 in Asia. It's missing. So why is that? Is the biology different in East Asia? Seems to us unlikely. Is this just a random effect because they happen to have this genetic variant in Europe and they don't have it in East Asia? We don't know the answer to that yet. All we can say for definite is that we do find differences between populations. It's not always true. Of course, there is some overlap, but it's not 100%. The next point I really want to come down to bring all of these things together is to ask, what do we know from this about the genetics behavior? So in summary, what have we found out? So the first point is that there are many genes. Now, I've told you that these studies don't tell us about genes. That's true, but they at some point will be related to genes. So if I go through and count up all of those places in the genome, the Manhattan plot peaks, how many are involved? And the answer now is pretty clear. It's thousands, thousands and thousands. And that, of course, is consistent with what I said about there being a very small effect attributable to each one. Since, say, the genetic contribution to schizophrenia is large, so the heritability is something like 80%. Almost all of the risk of schizophrenia is genetic, but the individual components of that are small, and therefore there has to be a lot of them. We don't really understand yet what that means. Lots of arguments have been going on about how we interpret that finding. Does it really mean half the genes in the genome are involved? And if so, how can this be specific? Because half the genes in the genome are not involved in making us mad. They're doing lots of other things. So how can this be explained? This remains a conundrum for geneticists. And as I pointed out, each makes a tiny contribution. So these 2 points really go together. There are many genes and each makes a small amount. And then the last point, and this is the last slide, is that we have no idea how these genes have their effect at the moment. We do know for conditions like hypertension, for diabetes, there's a lot that these studies have begun to tell us. But for behavior, we are really only at the beginning of this research. We have very little idea about what these things will be doing to make us mad, to make us depressed, to make us anxious. And that enterprise, trying to sort out that puzzle, is going to take us many, many years to sort out. And perhaps in another 5 or 10 years, I can come back and we can have another discussion about what the new research has told us around those questions. So thank you for your attention, and I'm willing to take any questions.
Okay, thanks, Jonathan. Yoka has a question. Want to ask your question? Unmute. There you go.
I was just wondering, but you started to answer it right after I asked a question. I was wondering whether you found any consistencies that showed a genetic component to national character. Like the Germans are very orderly and obsessive, let's say. The Italians are much more relaxed, let's say, and so on.
So we can do the following. If I had from this group genetic information, I could reasonably well predict which country you originate from. I can tell you something about your ancestry. And I can also, to some extent, tell you a bit about your personality. As you've seen, we've got a bit of information about the personality trait neuroticism. But unfortunately, we don't really see any correlation between those two. So I'm not sure we could address that quite in the way you've suggested. I suspect that's probably because the cultural effects are larger than the genetic effects. There may be something there, but we don't yet see that.
Thank you.
You're welcome.
Naomi had a question.
Yes, it regards epigenetics, and coming at it for two reasons. One is a personal one, in that my mother was traumatized by the Holocaust, and I also read some articles some years ago about studies in mice in which mice that had been traumatized by electric shocks in association with a smell, their children and grandchildren showed anxiety, even though they had not been traumatized when exposed to that scent. And I'm wondering specifically if there are any studies on the inheritance, not through environmental reasons, but genetic ones, epigenetic ones, maybe through methylation of trauma, genes associated with that. Especially nowadays, I'm concerned about it because all the children in Palestine growing up traumatized, and if they will pass it on to their children and grandchildren, and wondering about Israelis themselves, if part of their, like Yoka mentioned, national trait being very concerned with security above everything, if that is partly genetic.
Yeah. So yes, many people have been looking at this, and they've also looked at specifically in Holocaust survivors and the children, and indeed the grandchildren. Unfortunately, those studies are a bit like the ones I presented you at the beginning of my talk, where some people find one thing and other people find another. And the likely interpretation is that because this is a very complex phenomenon and there are many things that are contributing to it, we'd need a very large sample size, and we'd need to be very careful about how we did the analysis. So all at the moment, I think we can say is that it's a good hypothesis, but we don't have the definitive evidence to say one way or the other.
Can you mention some people who are studying this?
Yes, I can give you, do you want the names? I can maybe send them to you. Or
Yeah.
okay.
You could put it in the chat.
Yeah, I'll look them up. Yeah, okay.
Thank you.
Sarah? I have a personal question besides thanking you for this fascinating discussion and all this information. It's I detected some humor in there, and I was curious about whether psychiatry or genetics came to you first. How did you get from one to the other?
So I had an initial training in molecular biology, in fact, in hematology. And I was working with a group who were looking at an unusual genetic cause of anemia. And it turned out that the people with this anemia that they were investigating also had some form of intellectual disability. Didn't quite understand why. And it turned out this was because of a chromosomal abnormality, which was hard to detect. And I spent some time working with that group, and we came up with a technique which would allow us to identify those chromosomal abnormalities. And while doing that, it occurred to me that we could start applying the same technology to cases of severe mental illness, because that really hadn't been looked at. So the answer to your question is that genetics came first.
Thank you. David?
Yeah, I want to repeat Sarah and saying thank you for this talk. It was really fascinating and totally enjoyable. And just to address Sarah's second comment about she detected some humor there, molecular biologists are very funny people, by the way. So I just wanted to make that statement. Anyway, I was wondering, you found through GWAS all sorts of different associations with different diseases, and you pointed out that the population differences between the Asians and the Europeans were very dissimilar. But to kind of narrow down some more likely candidates for a cause and effect sort of thing, were there any striking similarities between the different populations?
Yes, there are, and there's a big initiative at the moment to try and collect information from lots of different populations to address exactly that question, because we should, it's sort of like triangulating on this problem, because you'll find, supportive information from one which you find is in another would really help you deal with this problem I mentioned earlier about how do you find the genes, because we have this difficulty, we're not getting at the genes, but if we were able to combine information from different populations, then potentially we could do that. Those studies are still in process. There are some where this has been applied for things like obesity, diabetes, just beginning to for behavior. Those will be coming out, I think, in maybe 2 or 3 years.
Yeah, great. Thanks.
You're welcome.
I have a question about what it's like doing that kind of research in China. You made it sound very smooth and easy, although a lot of hard work, but I just wonder about government restrictions and sharing of data. And also your biography said you had worked in what I assume is South Korea. It just said Korea in the bit that Joe sent us. And the comparison between working in South Korea and China.
Oh, just always work in South Korea. It's so much easier. They're so much more organized. And yes, no, it proved really hard. I mean, the major reason we moved to South Korea was because it was proving impossible for the reason you mentioned about government restrictions and data sharing requirements. Particularly when we first started in 2007, it was a relatively open environment and a lot of enthusiasm. People were being welcomed. Hospitals would, basically say yes if we asked them to do things for us. And over the 7 or so years that we were doing this work, it got increasingly oppressive under Xi Jinping. Yeah, that's a shame.
Yoka has a follow-up question. You unmute?
I was wondering whether there was any thought yet about how this knowledge could be used to treat mental and emotional conditions. I mean, is there such a thing as what is it, gene something wrong?
Yeah, so of course I get funding from grant giving agencies, in this case from NIH, and when I write those grants, I always say we're going to do this study so that we can cure schizophrenia, cure depression, and then we do these big studies, and then we're looking at these results and we're thinking, are we any closer to this? In the longer term, we'd hope to understand what the biological mechanisms are that underlie these conditions, and then being able to use that to design treatments. But for psychiatric conditions, there's been very limited progress. But to give you a positive example, I think this week or the week before, there is a paper doing exactly the same thing, but for inflammatory bowel disease. And they do genetic analysis, and they find some bit of the genome, not a gene, but they manage to take that information and, as it were, turn it into a gene. And then they can use that information to say, well, what is this gene doing? And it turns out it's involved in determining inflammation in a particular type of cell, macrophages, in the gut. And they work out what the biochemical pathway is. And then they go and test a whole series of compounds which are used for other conditions. And they find that some of these compounds inhibit this pathway. And then they go and test that, and they find, yes, this seems to reverse the inflammation. So I'm thinking, yes, that's really good. That's just what we want to do for depression and for schizophrenia. Whether we can, well, we'll see. So I mean, that's the longer term hope, but it's proving a lot more difficult than many of us imagined.
Thank you. And thank you for your talk.
Not at all.
Any more questions? Okay, go ahead.
I know someone who, she's 24, and she tells me that she's the 3rd generation of her family who suffers from a kind of uncontrolled depression, whatever she's tried. And I'm just wondering, how would someone, so you're saying that they would see how the gene or chromosome is causing it, and then try to give a drug, for example, that would counter that. but how would someone in her situation track the studies and so that she could really be completely up to date on anything that might help her?
So I have to say we've not been very good about this, but so I'm a member, a leader of what is called the Depression Grand Challenge at the at UCLA, and we are supposedly coordinating, and we should have on our website exactly answers to these questions. But I'm embarrassed to say I don't think it's quite as up to date as it should be. But that would be one place to look. But at the moment, we haven't really got anything to show for all of this work in terms of actually helping our patients. I'm hoping 5 or 10 years. I give talks and I say things like, by the year 2040, we'll have reduced the burden of depression. I'm thinking, well, that's great because I'll have retired by then, so you won't have to shout at me when it's not true.
Thank you. Very interesting. I love knowing how you try to determine if you have found something that is, significant enough.
Big problem.
Joe, you have a question?
I do. First of all, Dr. Flynn, again, thank you for the presentation today. We in our group are fresh off a talk about artificial intelligence. Of course, you talked about what you've done over the last 10 or 20 years and your peers on meta-analyses, which kind of sort of is beginning to look like, a kind of artificial intelligence answer, in that metadata is used. So what are you going to do with artificial intelligence? Is this a new possible tool for you in some way?
So it's so the big advantage of artificial intelligence, it recognizes patterns that other methods cannot do, which is great because for things like voice recognition, and we've been using it for trying to work out whether people are depressed from recordings. We're thinking of deploying it in what we call digital phenotyping. So we've been collecting patients who've got an app on their phone which reports back to us how much they move around, what time they go to sleep, and so on. And that's a lot of information, and we can use that to try and identify those patients who we think are going to get depressed, and then the idea would be we can offer existing treatments to try and prevent the recurrence of an illness. So we use it for those sorts of issues. The drawback, as you probably know, is that no one knows how it works. So we can recognize a pattern within our recordings, but exactly what those features are and how it's ended up at that, as it were, diagnosis is a little hard to sort out. So if you were trying to understand how is it that the brain interprets sound, how do you at the moment take the sound waves I'm producing and turn them into some meaningful construct? You could use artificial intelligence to do the same. It would produce words for you, but you wouldn't get any indication of what the actual mechanism was behind it. So I'm a little wary about how much we would be able to use it to answer more fundamental questions about mechanism so that we could go ahead and design drugs on that basis. It's going to take a bit longer before it gets to that stage where it can really give us those sorts of insights. But yes, I mean, there are many applications for it, and we think a lot about how we might apply it.
Thank you. Any other questions?
Yes.
Okay.
I have a question. If you ever figured out a way to turn off the chromosome that causes, say, schizophrenia, is there a danger of some sort of ripple effect throughout the gene?
Do you mean it might have side effects?
Yeah.
Yes. And so the answer to that is that we look for as it were, the opposite of schizophrenia. I mean, let me give you an example where we know this works. So there's groups that were looking for people who just ate hamburger after hamburger, steak after steak, and never had a heart attack. And it turns out that's inherited. And more than that, it turned out that there was a gene involved which was inactivated in the same way that you were talking about, say, turning off the chromosome. In other words, if you turned off that gene, you stopped people from getting heart attacks when they ate lots of hamburgers. And that meant that you designed a drug which would turn off the gene, you'd have the same effect. And the big advantage of doing it from the genetics is because when you look at these people who didn't have this gene and ask, well, what else is wrong with them? You wouldn't find anything. They had no side effects. And therefore you knew if you gave them the drug, which had the same effect as knocking out the gene, it would be safe. And that has proved to be the case.
Thank you.
Welcome.
If there are no other questions, it's 4:15 now, so let me just thank you from all of us, Dr. Flint. We promise not to hug you to thank you, but just to give you a little applause.
Yes, I'm safe from the hugs being on Zoom. It doesn't
there you are.
restrict the physical contact, so that's good to know.
Okay, so once again, thank you all, especially Dr. Flint, and we will see you all next month.
Thank you for having me, and thank you for your
Thank you.
questions.
Thank you.
Bye-bye.